Hello, students. Today we are going to discuss about principal features of development. Let's see first introduction. Each animal passes through similar stages of development. The stages between fertilization and hatching are collectively called embryogenesis. A great variety of embryonic types exist throughout the animal kingdom. But most patterns of embryogenesis are based on variations on 7 fundamental processes. These processes are fertilization, cleavage, gastrulation, organogenesis, metamorphosis, distinction between germ and somatic cells, and gametogenesis. Now we will see principal features of development. First feature is fertilization. It involves fusion of mature sex cells, sperm and egg. Sperm and egg are collectively called the gametes. The gametes fuse. It stimulates egg to start development and initiates a new individual. Fusion of gamete nuclei forms genome of embryo. Genome is collection of genes. These genes instruct embryo to start development similar to their parents. Second feature is cleavage. Cleavage is a series of much rapid mitotic divisions start immediately after fertilization. Zygote cytoplasm has enormous volume. During cleavage, this cytoplasm is divided into numerous smaller cells called blastomeres. Blastula is formed at the end of cleavage. Blastula is a sphere of blastomeres. Third feature is gastrulation. The series of extensive cell rearrangements after bastulation is called gastrulation. Now the embryo is in the gastrula stage. Gastrulation forms 3 embryonic germ layers. These layers interact and form organs of the body. Fourth feature is organogenesis. The process of formation of tissue and organs from germ layers is called organogenesis. The cells of germ layers interact with one another. They rearrange themselves to produce tissues and organs. This process is called organogenesis. Many organs contain cells from more than one germ layer. In some cases, outside of an organ is derived from one layer, but its inside is derived from another layer. For example, the outer layer of skin, epidermis, comes from the ectoderm. But it's the inner layer, dermis, comes from the mesoderm. During organogenesis, certain cells undergo long migrations. They migrate from their place of origin to their final location. These migrating cells include precursors of blood cells, lymph cells, pigment cells, and sex cells. Fifth feature is metamorphosis. Organism hatches from egg or it is born. In many species, it is not sexually mature. Such organisms undergo metamorphosis. Then they become a sexually mature adult. In most animals, the young organism is called a larva. Larva may look significantly different from the adult. For example, larva of silkworm moths does not have mouthparts. It cannot feed. Hence this larva must eat enough. Hence, the adult has stored energy to survive and mate. Most female moths male as soon as they enclose from their pupa, they fly only once to lay their eggs. Then they die. Sixth feature is distinction between germ and somatic cells. The separation of somatic cells and germ cells is one of the first differentiations. It occurs during animal development. A. Germ cells. In many species, a group of cells get separated. They produce next generation, rather than forming the current embryo. These cells are precursors of the gametes. The gametes and their precursor cells are collectively called germ cells. They are reserved for reproductive function. B. Somatic cells. All the other cells of the body are called somatic cells. Seventh feature is gametogenesis. Germ cells then migrate to the gonads. Here they differentiate into gametes. The development of gametes is called gametogenesis. Gametogenesis is not completed until organism has become physically mature. At maturity, gametes may be released. They participate in fertilization to begin a new embryo. Senescence and death, the adult organism then undergoes senescence and dies. Its nutrients support embryogenesis of its offspring, its absence lowers the competition. Thus, the cycle of life is renewed. Now we will discuss all these principles through a diagram in the life cycle of frog. The first step is fertilization, in which egg and sperm fuse to form a zygote, then zygote undergo cleavage and form a blastula, then gastrulation occur and blastula convert in gastrula, organogenesis in gastrula change it into an embryo. The embryo undergo development and a tadpole forms. Tadpole undergo metamorphosis and an adult frog forms. Bye bye students, I hope you understood in very well.